In this video, we're going to be talking about content from section 3.3 and in chapter 3, talking about cells. So here we are with the eukaryotic cell. Remember, on the outside of the cell, we said there's a plasma membrane, also called the cell membrane, and that's the boundary between the inside and the outside of the cell. In this picture, the plasma membrane would be the barrier, again, on the outside of the cell, allowing the inside of the cell to look different than the outside of the cell. So the next learning outcome says describe the structure and function of the plasma membrane. So this is a tremendous enlargement cartoon of that plasma membrane. So they're showing you that's the membrane on the outside of the cell. Remember, the plasma membrane controls what goes in and out of the cell. So it says it's made mostly of phospholipids. And when we talked about chemistry, you learned a little bit about the group of organic macromolecules called lipids. And one of the group were phospholipids. And it turns out, remember, one of the hallmarks of lipids is they don't dissolve in water. And these blue balls with the strings on the end, each one of those is a phospholipid. So you can see that the membrane's made mostly of phospholipids, but it's in two layers. And those layers kind of form naturally. If here's the inside of the cell and here's the outside of the cell, cells are in aqueous environments. They're in environments that contain a lot of water. And the cytoplasm inside the cell contains a lot of water. Water is essential for life. Did you ever hear, like, on space things, they on reports about space, and they're looking for life on other planets, and one of the things they look for is water, because life as we know it can only survive in water. So there's water outside cells, there's water inside cells, and remember, lipids don't dissolve in water. With phospholipids, it's particularly these tail parts that don't dissolve in water. So the tails naturally orient themselves towards each other. They're hydrophobic, and the heads are going to orient themselves towards the inside or the outside of the cell. And this provides a layer of lipid that doesn't dissolve in water. And so it prevents the cell from dissolving in its surroundings. The other things you'll notice about the cell is there's a lot of proteins. And we said proteins are important structural molecules. So all these orange things are proteins in the cell. And the proteins can act as transporters. They can be receptors. And there's different things. But cell membranes have a lot of proteins. And they also have cholesterol. And we talked a little bit about how cholesterol is important for cell structure. If you look at this, remember the cholesterol, those 4 flat rings? They're also in the lipid group. There's some cholesterol, there's some cholesterol. And in the plasma membrane, what the cholesterol does is it kind of stabilizes the membrane. It stops these phospholipids from floating around so much, and it just gives it some stability. But it's essential for the structure of the membrane. Remember, we said that it's important for babies to have cholesterol in their diets. You don't want to give a baby a low cholesterol diet, because they actually need cholesterol to make membranes. Finally, see if you remember the function of the plasma membrane. It's going to control what goes in and out of cells. And that's why we're talking about it, because that can be pretty important. The next learning outcome says explain how some molecules move across a membrane by diffusion. And you might already know the concept of diffusion. You experience it. I think about it when there's somebody wearing perfume. And if you're far away from them, you might not smell the perfume, but if you're up close, you do. And so the perfume's in the high concentration on their body, and the further away from you they get, the perfume's kind of in lower concentration. And so the process of diffusion means the movement of molecules from high concentration until low, until they reach an equilibrium. So here's just a good visual example. It's a glass of water with the blue dye. And you put a couple drops of blue dye in there. And that blue dye is going to diffuse. It's going to spread from high concentration to low. Here's the low concentration, here's the high. It's going to spread naturally until it's uniform all over and it's in equilibrium. So the same thing about somebody wearing a lot of perfume. At first you only smell it when you're close to them, but after a while that perfume's going to spread out through the room and you might be able to smell it uniformly through the room if it's strong enough. In cells, with the kinds of molecules that are going to diffuse are going to be molecules that can easily pass through the membrane. So remember we said that phospholipid membrane or the plasma membrane controls what goes in and out of the cell. And some molecules can just naturally pass through the membrane pretty easily. Usually they're small, they don't have a charge, and they can pass through the membrane. And so let's say there's something on the outside of the cell. There's a lot of something, I don't know. And gradually, if it can pass through the membrane, some's going to move in until you have the same concentration on both sides. So we call that kind of movement diffusion, whether you're just in a glass of water, you're in the air, or it's going through a plasma membrane. Movement of molecules from high concentration to low until they're in equilibrium. No energy required. This just happens randomly because molecules are kind of bumping into each other. Molecules that can pass through the plasma membrane can diffuse, so they can move on either way. So like oxygen is a really good example. Oxygen's O2, it's a small molecule. It can pass through the membrane. So if there's a high concentration outside, eventually enough molecules will diffuse in that you'll have the same concentration of oxygen on both sides. Oh, I missed an H right there. The next learning outcome is explain how some molecules move across a membrane by osmosis and explain the concept of hypotonic, hypertonic, and isotonic conditions on their cells. For some people, osmosis can be challenging. It is a concept that is important every minute of our life. Our body has to be dealing with osmosis. If you understand osmosis, it might help you understand some physiological things in the body a little bit better. It's a really important concept. If you are pursuing healthcare and nursing or EMT or anything like that, you definitely want to know about osmosis. So put the effort in to understand this, because once you do, it's a pretty important concept that you can apply. So I talked about diffusion because I wanted to use that term when we described osmosis. Osmosis is the diffusion of water across a semi-permeable membrane. Semi-permeable means some things can pass and some things can't. So remember when we looked at our plasma membrane, we said some things can pass through here and some things can't. The plasma membrane is going to be what makes the inside of the cell and the outside of the cell look different. So osmosis is the diffusion of water. When you talk about osmosis, we're not talking about the movement of anything else but water. We're not talking about oxygen or CO2 or glucose or anything, only water. Osmosis, diffusion of water across a semi-permeable membrane. Remember, diffusion happens until you have the same concentration on both sides. So osmosis is going to happen until the concentration of the solution is in equilibrium. So let's get that vocabulary straightened out. When we talk about a solution, we talk about something like salt or sugar, because cells have a lot of salt and a lot of sugar in them, and that's called the solute, dissolved in a liquid, which is going to be water in the human body, and we're going to call that the solvent. So we're going to have the same concentration of solution on both sides of the membrane. Here's a video, an image they try to help you understand that. If this is a semi-permeable membrane, so this is a beaker. This might be something somebody set up in a lab. And they put a membrane here. They put some kind of barrier here. That if this is some molecule, some solute, the purple circles are solute, and the pink is a solvent, that's water. The purple things can't get across that membrane. They're too big. See, they won't pass through those holes. But the water can move by osmosis. And the idea is is this solution, the water's going to move until we have the same concentration of solute, the same concentration of the purple dots on both sides of the cell. If you look at the starting situation, we can see that the right side, the purple dots are a lot more concentrated. Your solute is more concentrated. Osmosis says water is going to move from high concentration to low. It's going to diffuse. So in this situation, the water's in low concentration relative to the solute. In this one, the water's in high concentration relative to the solute. So the water's going to move from high concentration to low concentration. And so this is showing you after time, the water has moved over to this side. And now the concentration of the solution is the same. And notice that it actually can create a pressure gradient and make the volume increase. And that's important in functioning of the human body. So diffusion, molecules moving from high concentration to low. Osmosis is diffusion of water. So this water moved from high concentration to low until you reach an equilibrium, a same concentration on both sides. I wanted to make a note that under your lecture videos in Brightspace, the last thing I have there is a video that I labeled as osmosis. I also refer to this same video in the lab handout. I highly recommend that you look at this video to understand osmosis. Okay, so that'll give you a little bit more detail to understand a little bit more. I'm going to go on to the next step, but that will kind of summarize what you would see in this video, but I highly recommend you watch this osmosis video too. Okay, so if you kind of get the idea of osmosis, we're going to be looking at human cells. And in this case, let's just say it's a blood cell. Okay, and this looks like a blood cell because they have this little dent in the center like that. If we put a cell in an isotonic solution, iso means same. So a solution that has the same concentration of solute, that means the same concentration of salt or sugar, whatever we're talking about, inside and outside of the cell. So here's the example where this cell is in an isotonic solution. These little dots are in the same concentration, that salt or sugar is in the same concentration outside the cell and inside the cell. Remember diffusion/osmosis, the molecules move until they're in equilibrium. So if the water is the same concentration on the inside and the outside of the cell, if a molecule of water moves out of the cell, another molecule is going to move in. And the cell is going to look like a normal cell. All of our cells are in an isotonic solution in our bodies. All that balancing of drinking and urinating and balancing your water and your salt that your kidneys do is to keep your body solutions, all the liquids in your body, primarily your blood, isotonic to the cell concentration. We want the same concentration inside and outside the cell. If a cell is in a hypertonic solution, that means hyper means high, and the thing that's high is going to be the solute. So these little dots, the sugar or the salt around the cell is going to be high. Let's say you're dehydrated or you've been eating things that are really salty. Your fluids in your body can get salty relative to your cells, and you can start creating a hypertonic solution. What that means is the solution around the cell has a high amount of solute compared to the inside. If there's a high amount of salt, let's use salt as our example, there's a high amount of salt outside of the cell, that means the water concentration is going to be lower. And if that seems kind of confusing, that's what I made that extra little video about. So go watch that extra video. But in this example, they're showing you the salt concentration is high outside the cell. That means the water's low compared to inside the cell. So if the water concentration's low outside the cell, it's higher inside the cell. Osmosis says water's going to move from high concentration to low concentration, which is why they're showing you a big arrow. Water's going to move out of this cell, and the cell's going to shrivel up. And that's not good. That will kill the cell. So we can't survive without enough water or with too much salt in our system. And that would put the cell in a hypertonic solution. If we took a human cell and we put it in a hypotonic solution, that's when the solution around the cell has a low amount of salt or sugar. And so here they're showing you that the salt concentration around this cell is very low compared to inside. That means the water concentration is high. The water concentration is high outside compared to inside. Osmosis says water is going to move from high concentration to low. So water is going to move into the cell. Just like we saw here, the water moved from this section of the beaker over here, and the volume is going to increase. If we increase the volume of a cell, that cell can enlarge, and it can ultimately pop if it's in a solution where the water's too high. So again, all that, eating the right amount of salt, urinating, drinking the right amount of water, all that's trying to keep our body fluids isotonic, so our cells don't shrivel up and so they don't burst. You put in enough water, that plasma membrane is not that strong. It's going to burst the cells open. So we call that a hypotonic solution. Most people get the idea of isotonic. That's like a normal solution, the cell's in its normal shape. Hypertonic solution is going to cause the cell to shrivel. Hypotonic solution is going to cause the cell to enlarge. And you get to a test and you start going, oh my gosh, hyper, I can't remember. Hypo has an O in it. Hypo. Think of that the cell gets big and round like an O and can potentially pop. Here's a picture with, these are red blood cells taken under a microscope like the one that we see in our lab, in a normal teaching lab. So this is easy for you to see under the microscope. See if you can figure out which of these cells are in an isotonic solution, which are in a hypertonic solution, and which are in a hypotonic solution. These are isotonic. These are normal red blood cells. They're round. If you put them in a salty solution, a hypertonic solution, they're going to shrivel up. And they can't survive that. That's not good. This happens all the time. Students will look at this under the microscope and go, wait a minute, there's nothing there. What happened? I can see a few things, but where'd all the cells go? I don't know. I must be magnifying it wrong. I must not be focusing right. But you remember when they're in a hypotonic solution, the cells can burst. So most of the cells in this preparation exploded. So you might see pieces of cells. So these would be the cells in the hypotonic solution. So like I said, go look at that video that's labeled as section 3.3 osmosis, and work on the osmosis part in lab and try to get this concept down.